Really excited to announce Dr. Irene Newton as our speaker this week, who is an associate professor of biology at Indiana University for her talk titled Mi Casa es Su Casa, How an Intracellular Symbiont Manipulates Host Cell Biology. The Newton Lab broadly studies host-associated microbiomes with specific focuses on arthropod hosts and their microbiomes and their endosymbionts. They leverage genomic, microbiological, and cell biological techniques to functionally characterize these host microbe interactions. And with that, I will turn it over to you, Irene.
Thanks, Killian. And thanks everybody for your attention. Since this is going to be recorded, I'll try my best to make it be clear and understandable later. All right, I'm going to share my screen. And I'll keep in mind that I actually can't see you guys for some reason when I share my screen. So if you have a question that just can't wait, just speak up, or you can put it in the chat and maybe Killian can keep track of that. All right, so as Killian mentioned, the title of my seminar is Mi Casa Su Casa. And the reason is that for these intracellular bacteria, their house is the host, right? So mi casa su casa means make yourself at home. You know, my house is your house. And these bacteria indeed do make themselves at home. What you see on the right is a dissected ovarioles from Drosophila melanogaster. So these are tissues in the ovary. And what we've done is stained for Wolbachia in green. The first thing that you should gather from this image is, oh my gosh, there's so many Wolbachia, right? Like it's insane how densely colonized this tissue is. Every single puncta that you see is a bacterial cell. Of course, if you had this type of infection in your gonads at such density, it would not feel so good. But amazingly, if I were to show you infected and uninfected flies, you wouldn't be able to distinguish them, and the over tissue neither. And this is not a phenotypic burden on the fly, and in fact can be a fitness benefit. But the fact that these infections are persistent and that they're not cleared suggests certainly that they are manipulating the host biology to be able to persist, to replicate, and to be transmitted to the next generation. Now, I know this is a broad audience, and so I thought I would begin with the big picture. And I can't think of a bigger picture than this one, our planet, the Earth. And it'll give you, just a brief history of our planet. About 4.6 billion years ago, it formed. Shortly thereafter, microbes arrived on the scene. And through their amazing metabolic capabilities, they dramatically changed the geochemistry of the planet, producing increasing quantities of oxygen. And this is something, that as we take breaths today, we need. But of course, it continues to be pretty toxic to many organisms. And this selective pressure is what's thought to have formed the first symbiosis that we know of, that of the eukaryotic cell. When an ancient unicellular organism, ancestor to the mitochondrion, able to use oxygen as a terminal electron acceptor, was taken up by another unicellular organism to form this union of two different organisms, differently named, living together, which is really the definition of symbiosis. Right, so if we fast forward to present day, Metazoa arrived just under 1 billion years ago, and we are a tiny blip on this evolutionary scale. And what I hope you get from this is that we evolved in a microbial world, and we are living in a microbial world. So it should come as no surprise that the interactions that we have with microbes have had substantial and really dramatic effects on our ecology and evolution, and they continue to do so today. So it's really important to study the mechanisms of symbiosis, not just because it's cute and I think it's interesting, and it's foundational to all of eukaryotic life on the planet, but also because eukaryotes continue to form these types of interactions, and they can really alter the evolution and ecology of those organisms. So I'll give you just a few really emblematic examples. So first, on the left, you see the giant tube worm Riftia pachyptila. Riftia lives at the bottom of the ocean near volcanic hydrothermal vents. Using the chemical energy from these vents, that of reduced sulfides, symbionts that live inside of this tube worm in a special organ called the trophosome fix carbon dioxide for their hosts. The hosts are mouthless and gutless and couldn't live here at all, except for the presence of their symbionts. So they're kind of like plants living at the bottom of the ocean using chemical energy instead of sunlight.
Hey, Irene.
Yeah.
It sounds like you have a new slide on the screen, and I still only see your first one with the history. I don't know if that's just me or others in the audience.
This one? Uh-oh. It's not advancing?
It seems like maybe it's not advancing. Yeah, It looks like others are seeing the same thing. Maybe you want to like unshare and then read? Yeah.
Yeah. Sorry about that. Thanks for letting me know. Sometimes I think Zoom does that if you're not sharing. All right, so were we here? Can you see anything now?
I'm not seeing it. It said you've started screen sharing, but I don't see anything.
Not yet. Okay.
Zach has a comment in the chat about dual monitors. Which is likely.
Let's see.
Maybe the issue, but.
So let's see, what does the chat say? Okay. It happens if you use 2 monitors. Yeah. So I can try to unplug one of the monitors. Let's see.
Sometimes it works too if you just use the window that the program's.
That's what I'm doing. Yeah. Got kind of like a complicated setup, but I can definitely unplug one of them. All right. So if I unplug one of them, then we'll try again. Sorry about that. Stop share. What about now?
I'm not seeing any slides on. That's really weird.
You try sharing the screen, not just the window also?
That's what I'm doing. Yeah, I'm just sharing the full desktop. And I've tried sharing just the keynote. That's just the keynote.
Oh, weird.
Do you have a PDF you can open?
I don't know why that would make a difference, because if I can't share the desktop, I can't share anything.
Oh, yeah.
Could it be something with the actual... Oh, wait a minute. I see something now. I see the desktop.
You see a desktop, but do you see my talk?
Yeah, but you're about to play it. I see the mouse.
Now do you see something?
Yeah, do you want to try and advance?
Okay, we'll try advancing. Yes.
Okay, microbes.
Okay. Yes. Oxygen.
Yep. See the animation.
So now you guys see the animation that maybe you didn't see before. Okay, so big picture, we're living in a microbial world. It's important to study symbiosis and study systems. Okay, so now we're back where we were before. So I mentioned Riftia pachyptila and its amazing symbionts. And on the right, you see Buchnera aphid symbiosis. So the aphid survives on this diet of nutrient-poor plant sap, really like low in amino acids, because it's got this symbiont Buchnera that is able to provide them to supplement this nutrition. And the tsetse fly, similarly, you might think that blood is very nutrient rich, but it's actually vitamin deficient. And so its symbiont, Wigglesworthia, provides it with vitamins that are missing in the blood. So in each of these cases, the bacterial symbiont has allowed the host to occupy a niche and flourish where it otherwise wouldn't be able to. But also in each of these cases, we know very little about how these symbioses are formed and maintained. We don't really understand the mechanistic detail of how these organisms interact with each other. And enter Drosophila melanogaster. In my lab, this is the model system we primarily use. It's definitely not as sexy as hydrothermal vents, but it's enormously tractable. And the symbiont that we study is Wolbachia. And for those who haven't heard of Wolbachia before, it's the queen of the alpha proteobacteria. It infects 40 to 60% of insects on the planet, so insect species, as well as arthropods and filarial nematodes. So really broad spread intracellular infection. And it was primarily initially known for being a reproductive parasite, for doing these things called cytoplasmic incompatibility, which is where sperm and egg are not compatible anymore, so limiting gene flow between populations. It also does more curious manipulations, such as male killing or feminization or parthenogenesis induction. So it's kind of the more, I guess, outlandish Wolbachia phenotypes. And it's all because Wolbachia wants to benefit infected females, because it's the infection that keeps on giving. And as I showed you in my first slide, Wolbachia densely colonizes the reproductive tract of females, and it's able to position itself to be really efficiently maternally transmitted. So those eggs come preloaded with their Wolbachia very early on. And once they're laid and fertilized, the organism is developing in the context of that infection. Wolbachia, in 2008, was discovered to also confer this important benefit to arthropod hosts in that it prevents RNA virus replication. So it may be one answer to control infectious disease vectors. And I'll explain why. So this phenotype, where Wolbachia protects insects from virus infection, was discovered in melanogaster. And that bacterium, W. mel strain of Wolbachia, was moved into the important human vector Aedes aegypti. And these mosquitoes will released all over different places under the premise that if they take a blood meal from a person who has dengue, for example, or Zika, the virus can't replicate inside of the arthropod. So then when that mosquito goes on to bite an uninfected person, it can't transmit the disease. And this is not just purely academic. There are many different places all over the world where these mosquitoes are being deployed right now. This is a map from the World Mosquito Program out of Australia that has been very successful in deployments of mosquitoes containing Wolbachia in different places. And just now they're just getting data from these sites suggesting that indeed the Wolbachia infection has been successful in limiting the amount of dengue in that region. All right, so today's story is going to be one in 3 parts. So first, I'll tell you how this intracellular infection Wolbachia alters host biology using an RNA-seq approach. I can then describe the symbiont's toolkit. So how exactly is the symbiont altering host biology? Then I can tell you about how these changes protect from virus replication. So first, how does an intracellular infection alter host biology? So we're an omics lab, so we did take an omics approach, like have hammer looking for nail. So amazingly, the transcriptome of a Wolbachia-infected melanogaster fly had not been done until our project. And so we took Wolbachia-infected or uninfected age-matched female flies, and then we extracted total RNA with Trizol. We used terminase to remove the ribosomal fraction, and then prepared libraries for the with NDB-Next for the NextSeq instrument. So the question we're asking is really, how does host gene expression change in response to Wolbachia infection, and what might this tell us about cellular pathways that the symbiont manipulates? And this is the schematic of the workflow that we use once we have those reads. So the raw data is quality controlled. We use a program called RSEM to map the reads to the most recent assemblies for melanogaster and Wolbachia. We used TPM extraction tools and R to be able to then quantify differentially expressed genes. And then the advantage of using flies, well, there are many advantages of using flies, but one huge advantage is just the wealth of knowledge that we have for protein-protein interactions and genetic interactions as well. So we used something called the string network to ask of all of our differentially expressed genes, do they cluster based on known genetic or protein-protein interactions? That is, could we identify functional hubs based on all those differentially expressed loci? And what I'll show you in the next slide is that network that we produced. So this is very busy, so I'm going to spend some time walking you through it. First, every single node is a differentially expressed locus in melanogaster that was responsive to Wolbachia infection. If it's upregulated, it's in blue. If it's downregulated, it's in green. If these proteins or interact with each other, or if there's a genetic interaction between nodes, then there's a line connecting them. The more connections, the bigger the node is drawn, right? So like UBP63E here is bigger because it's like got a lot of spokes coming out of it. As part of the way that we sequenced this RNA, we were also able to get at differential isoform usage, which is something that nobody had previously shown for an endosymbiont. And so what I think is really fascinating is that many of the loci that were differentially expressed were also differentially spliced in response to Wolbachia. And that is shown by this little pink circle that goes around that node. And finally, we clustered these based on functional importance. So you can see Wolbachia impinges upon these really significant parts of host cell biology: stress response, ubiquitin, RNA binding and processing, metabolism, transcription and translation, recombination and cell cycle checkpoints. I thought it was particularly cute that we highlighted all this differential isoform usage, and then we also saw RNA binding and processing being affected by Wolbachia. And some of this we expected based on phenotypes that we already knew for Wolbachia. So for example, we knew that Wolbachia, based on genome sequencing, it's an auxotroph and amino acid scavenger. It relies on host proteasome, autophagy, and translation to be able to replicate. So our lab had done most of the genomic work on it. There were other groups who had done RNA experiments to be able to show that host translation initiation and ribosomal proteins are really key for Wolbachia titer in cell lines. We know that Wolbachia can alter perceived recombination in Drosophila. So your own Nadia Singh had made this discovery that Wolbachia infection increases perceived recombination in the fly. And we had followed up with a couple of other experiments. So it was nice to see that process coming up in our RNA-seq data set. We know that Wolbachia produces a really potent toxin that interferes with host reproduction, and that toxin contains a deubiquitolase. So here I'm just showing you a schematic at the top of this toxin that's involved in sperm-egg incompatibility, one of those reproductive phenotypes I mentioned at the beginning of the talk. We know that expression of the toxin alone is sufficient to induce this reproductive phenotype, these CYF-A and CYF-B genes. And so it's suggestive of Wolbachia altering host ubiquitin pathways. So this is kind of just a rough map of the things that Wolbachia might alter and how these processes might interact with each other. And I can tell you a little bit about how Wolbachia and its own proteins might actually enact these changes. And I can tell you how these changes protect from virus infection. But unfortunately, I don't have time to tell you both of these stories. So we're going to have a little choose your own adventure. And I don't know if we got the poll to work ahead of time. So do we have a poll?
Yep, I see it.
Okay, so you can choose whether you like to hear about your symbionts toolkit, or if you want to hear how we think the Wolbachia changes host cell biology to protect from virus infection. I don't actually see anything like going, so I don't know who can keep track of the votes.
I'm watching it, so we've got almost everybody. I'll give people like 10 more seconds.
Oops.
All right, well, I think, looks like it's going to be B.
Okay. It's
Viruses.
going to be B, viruses. You got it. All right, so we're going to go down here. Congratulations, you've chosen how do these host changes protect from virus infection. All right, so I don't need to convince anybody these days that RNA viruses are important disease agents, right? In this pandemic going on now, I think that's pretty evident. But arboviruses are a really important group of RNA viruses. And these are those that are transmitted by arthropod vectors, such as mosquitoes. Oh, for some reason, I'm seeing the pole in my face. Sorry. Okay. For arboviruses, there are no vaccines available, and there are limited treatment options. And so that means that vector control is the emphasized way in which that we control the spread of arboviruses. Now, I mentioned that Wolbachia limits virus replication, and I wanted to show the original experiments that were published that showed that in Drosophila melanogaster, its native Wolbachia protects from Drosophila C virus. This is a really important virus in the fruit fly that kills them. You see on the left is Luis Teixeira's paper showing that flies that were progeny of resistant mothers, so they inherited the Wolbachia, were all protected from DCV compared to those that weren't. On the right, you can see a similar paper from Jeremy Hedges showing that their survival rate is much lower when you have flies that are cleared of the infection with tetracycline. So comparing genetic controls. Now, how might Wolbachia change host cell biology to limit virus replication? So in this schematic, you envision Wolbachia in its host-derived vesicle, because that's how it lives inside the host cell. It's got a type 4 secretion system that it can use to pump all these effectors, different proteins into the host cell environment to change a variety of different things like vesicle trafficking. It can change the membrane composition. It can induce ER stress and reactive oxygen species. All of these different changes in the cell act as a syndrome, changing a variety of different things that might impinge upon virus replication. So the virus has to be able to come in through receptor-mediated endocytosis, dump its genome into the cytosol, make these little replication factories that rely on the membrane, make more genome, make more protein, and be able to leave the cell. So because of the way in which Wolbachia interacts in the cell, we might imagine that there's lots of different things that could alter the virus's ability to replicate. And indeed, there are many different hypotheses out there for how pathogen blocking works. Before I show you our data, I want to begin by explaining the model that we started to use and also highlighting the fact that this is a collaboration between my lab and that of Dr. Rich Hardy, who's a virologist here at IU. And we had two very talented trainees that worked with us in part of this project. Dr. Amelia Lindsay, who was a postdoc in our labs and went on to start a faculty position at Minnesota. And now Dr. Tamanash Bhattacharya, who was a co-mentored graduate student on this project, who is now a postdoc in Harmeet Malik's lab. So the system that we developed is that of the fruit fly and its native Wolbachia WMel. And then we use the alpha virus Symbis as our model alpha virus. And the reasoning was that Symbis is kind of the canonical model for alpha viruses. We have a lot of genetic tools in Symbis, a lot of genetic tools in the fruit fly, and maybe we can figure out exactly what's going on. But of course, we first needed to know if pathogen blocking would even work for Symbis, Wolbachia, Drosophila. WMel in the fruit fly has never seen Symbis virus, right? Of course, it makes sense that WMel would protect against DCV, but in this case, would we see pathogen blocking? So the sub-outline for this little talk is how we've developed this model, then how we identified host MT2 as responsible for virus inhibition, and how MT2 might act in pathogen blocking in mosquitoes. All right, so these viruses, these alpha viruses like Symbis, are positive sense single-stranded RNA viruses, and they act much like an mRNA, having a cap and a poly A tail. And what's fascinating is if you take these viral genomes and plunk them inside a eukaryotic context, they immediately initiate infection on their own. And as part of that, they go through a minus-strand intermediate, and then they make two different positive sense genome entities, one containing the structural polypeptides, and then the full-length genome that can go on to be encapsulated. We wanted to know if Wolbachia initiates pathogen blocking in the context of Symbis. And so the first thing we did was just measure copy number of genome replication in the presence or absence of Wolbachia. So W plus are flies with Wolbachia, W minus are those without. We looked at both plus and minus strand, and amazingly, we did see pathogen blocking. So Wolbachia inhibits Symbis replication in fruit flies, even though Wolbachia has never seen Symbis before. We can also measure the amount of protein produced in the system by using an eluciferase reporter construct. And so what you can see here is that relative to Wolbachia infected flies, Wolbachia free flies, we see a significant reduction in total amount of protein, both structural and non-structural proteins. But if we normalize for the amount of total RNA that's present, there's no difference, meaning really, the defect in replication is because Wolbachia is altering genome replication for the virus really early on. And if you have less genome, then of course, you're going to have less protein. So in the schematic of the virus life cycle, the virus has to get in through receptor-mediated endocytosis. It's got to dump its genome in the cytosol. That genome acts like an mRNA. It's acted on by host ribosomes, making these viral proteins that produce more genome. You get more viral proteins, you get the production of these encapsulated virions that can then exit the cell. And these progeny virions, even in a normal infection, some subset of them are not infectious. And I'm kind of shown those as having no color here. So we can actually measure the proportion of infectious and uninfectious particles coming out of an arthropod cell, kind of mimicking the transmission and transferring them to mammalian cells. So we generate these progeny virions in an arthropod context with or without Wolbachia, and then we overlay them on BHK cells and ask, about plaque forming units, how many can go on to make plaques. So that's what you see here. On the left are plaque forming
Irene,
units.
can I ask a quick question?
Absolutely.
Do the viruses need to have the RNA packaged in order to be actually secreted to be exocytosed, or do they
You might
assemble without it?
imagine, yes, you might imagine that some particles could be empty. In the case of Symbis virus, there are interactions with the RNA itself and the capsid proteins, and so largely that doesn't happen. But you could imagine that there are defective particles that come out that have maybe truncated genomes or different types of RNA entities inside of them. Yeah. Does that answer your question?
Yeah. Mm-hmm.
Okay. All right, so on the far left, you can see that we measured these plaque forming units in the absence of Wolbachia. You can see we can measure particle number, just looking at genome copy number with qPCR. And the ratio of these two is what we call particle infectivity. And the lower this number, the more infectious the virus. This is without Wolbachia around. And in the presence of Wolbachia, we see that a huge increase in this particle to PFU ratio. So suggesting that a lot of these particles that are coming out of Wolbachia infected cells can't go on to initiate an infection in mammalian cells. So this was an interesting phenotype. Another way of showing the same data is by showing the ratios a little differently in specific infectivity. So on the right, you see that about 60% of the viruses that come out of a normal arthropod cell can go on to form an infection. And we see a dramatic reduction in total particles that come out of a Wolbachia infected cell, but we see an even more dramatic reduction in infectivity. So the particles that are being produced just can't go on to form an infection. Another way of saying that is Wolbachia infection results in the loss of infectious virus particles. So in conclusion, for this tiny part of this second part of the talk, Wolbachia does inhibit Symbis. Our model works. We see that Wolbachia reduces virus genome replication very early during infection of an arthropod cell. It does reduce protein production too, but that's likely due to a reduction in genome replication, and that Wolbachia alters virus-specific infectivity, reducing the fraction of infectious virus particles. So next, we really want to know how Wolbachia does it. How is it that Wolbachia is limiting virus replication? And you might imagine that there's some direct antagonism, right? That the Wolbachia is secreting some protein that specifically antagonizes virus. But this is quite unlikely. As I mentioned many times, Wolbachia has never seen Symbis, and Wolbachia actually is effective against a huge array of unrelated RNA viruses. So it's quite unlikely that this 1 megabase genome contains components that specifically target each of these rapidly evolving entities. Instead, we believe that Wolbachia acts indirectly, antagonizing the virus through changes in the host cellular biology. Now, what I didn't tell you guys before, and I got hid from you when I showed you that RNA-seq data set, is that there was another important variable that we included, and that was Symbis infection. So I can show you now that we had RNA-seq data from flies with and without Wolbachia, and also with and without Symbis, and that we sampled them 6, 24, and 48 hours post-infection. The same kind of workflow I showed you before, including string network analysis, applies. And first, I'll show you kind of overview data. So these points on this graph are medians of all of our 5 replicates for each of these conditions and time points. And the shape of the point corresponds to the time. 6 hours is a square, 24 is a circle, and 48 hours is a triangle. The colors correspond to condition, plus minus Wolbachia or plus minus Symbis. And then the arrows are there to simply guide your eye from one time point to the next. And what I think is kind of cute is you can see in the conditions where we don't have Symbis, that is the blue and red conditions, that the fly starts out in a certain transcriptional space, it's poked, and of course then has an injury, and the transcription changes. And then as it recovers, it kind of starts going back to where it was before. But in the conditions where we have Symbis, it starts out in some transcriptional space and then just flies off into a different trajectory, never to come back. And that makes sense because Symbis virus, like all these other arthropod virus, would persist in the host and kind of stick around, changing the way that we perceive that transcriptional response. If we take these same data and we rotate them on their axis, I can show you that they cluster by Wolbachia infection and also by whether these flies were infected with Symbis. But what actually changed, right? One other way of viewing these data is by showing you a heat map. So each one of these lines or rows is a host gene that was responsive to virus infection. And each column is one of our experimental replicates. We've clustered the data based on time and also by condition, plus minus Wolbachia, plus minus virus. And the two sets of columns to compare that are most interesting here are these. 6 hours post-infection, so really early during the infection, in the absence or in the presence of Wolbachia, but both of these sets have Simbus. And what you can hopefully see is that when Wolbachia is absent, we see this pretty strong transcriptional response from the host. So lots of genes are upregulated and many are downregulated. However, in the presence of Wolbachia, the response is much more muted. It's almost as though the host doesn't really perceive the infection the way that their Wolbachia-free counterparts do. And that makes sense because, as you saw before, the virus isn't able to produce as much genome or as much protein when Wolbachia is present. Just as we did for Wolbachia, we can also make a nice string network for the virus. In this case, these are host genes that were responsive to virus. And I'm showing you all of the known genetic or protein-protein interactions between them. Just as before, the size of the node is really just indicative of the number of connections. We have a few genes that look like they have differential isoform usage in response to virus. And interestingly, we have two major hubs that are functionally important for virus infection. The endoplasmic reticulum, no big surprise, because the virus needs that to make these replication factories. And then metabolic processes, such as purine, sarcosine, and carbohydrate metabolism. But this doesn't tell us how, Wolbachia and virus impinge on each other, right? So we wanted to look for host genes that were differentially regulated by virus and Wolbachia, and host genes that might be associated with antiviral immunity. And that's what brought us to this gene MT2. So for those unfamiliar with MT2 or DNMT2, it's a methyltransferase and the lone methyltransferase in dipterins. It's known to make these 5-methylcytosine marks, and although it's canonically, I would say, characterized in mammals, where these enzymes interact with DNA, in dipterins, they interact with RNA. And they are induced following ER stress or reactive oxygen species. And at least one paper before we started working on this project in 2013 showed that MT2 is antiviral against Drosophila C virus. So if you overexpress MT2, it protects against Drosophila C virus. And we knew from our transcriptomic analysis that MT2 expression was elevated by Wolbachia infection, but repressed by the virus. So I'm showing on the right qPCR confirmation that when flies are mock injected, but have Wolbachia, we see an elevation of MT2. But when virus is around, this elevation goes way down. So suggesting that Wolbachia and virus are fighting over this important transcriptional hub. And just to convince you that this is not an artifact of the strains we are using or the fact that they have virus, in all of these flies I'm showing you here, infected with a panel of different Wolbachia strains, MT2 expression is increased across the board. So it's a common phenomenon for fruit flies who have Wolbachia to have elevated MT2. We then asked whether MT2 would be antiviral against Simbus in Drosophila. So we took flies without Wolbachia and overexpressed MT2. And even though we only saw a very modest increase in MT2 expression, we saw a dramatic reduction in viral RNA replication. And interestingly, also a drop in infectious virus titer. This is very similar and reminiscent to what we had observed for Wolbachia infection. We also took advantage of the fact that we have MT2 nulls to ask whether MT2 was important for pathogen blocking. So on the left, you see changes in viral RNA, and on the right, infectious titer. In the context of Wolbachia, if we knock out MT2, we do see an effect on the ability of Wolbachia to protect its host from virus replication. So without MT2, Wolbachia can't protect as well. Oops. All right, so as I mentioned before, specific infectivity is the way that we look at what fraction of these total particles coming from the infection, the progeny virions, are infectious. And without Wolbachia, but if we overexpress MT2, we see this decrease in specific infectivity. But if we mutate the catalytic residue important for the methyltransferase activity, now MT2 can no longer have this effect, suggesting that the methyltransferase activity of MT2 is important for this reduction in specific infectivity. So both Wolbachia infection and methyltransferase result in the loss of virus infectious particles and that methyltransferase activity itself is important for that effect. So in order for the methyltransferase to act on the virus directly, it would have to interact with the virus directly, the viral genome. So we performed a RNA immunoprecipitation experiment with flag-tagged MT2, and then used the RNA fraction that we could pull out of these cells as template for quantitative PCR. And what you can see here is compared to empty vector, we see an enrichment of RNA genome when we pull down with MT2, suggesting that MT2 does interact with the viral genome. All right, so for this part of the talk, I conclude that Wolbachia and Simbus kind of fight over DMT2 expression in the fly. I hope I've convinced you that many Wolbachia strains elevate MT2 expression and that virus infection reduces it. MT2 is antiviral for Simbus and Drosophila, just as had been shown for Drosophila C virus, and it's important for pathogen blocking. MT2 directly binds Simbus virus's genome, and the methyltransferase of that MT2 is important for reduction of infectious virus. So it leads us to a model where Wolbachia in the cell is upregulating MT2 expression through some unknown mechanism. The increase in the presence of this enzyme acts on the viral genome, perhaps in many different parts of the replication cycle. And this results in a loss of infectious particles as these progeny virions are produced in the arthropod. All right, but one thing that's I've heard at study section, and maybe some of you have too, is mosquitoes can fly, but flies can't mosquito. I think it's a pretty funny thing to say. It's true that flies and mosquitoes are both dipterans, but the biology of these organisms are really different, right? And so if we wanted to really learn about Wolbachia-mediated inhibition in pathogen blocking, we have to transition what we've learned in melanogaster to mosquitoes. So are the things that we've been studying in our model system really translatable to the important vector host? Does Wolbachia alter specific infectivity in mosquitoes? Is MT2 important for Wolbachia mosquito pathogen blocking? So first I'll show you that regardless of the virus or Wolbachia strain, specific infectivity of viruses is reduced in mosquitoes in the presence of Wolbachia. Here you can see for Simbus, Chikungunya, and Zika virus, in the presence of strain W-MEL, we see a reduction across the board in specific infectivity. The same is true for 3 different Wolbachia strains in the same mosquito background. Then we asked whether MT2 in mosquitoes would interact with Simbus, just as we had seen for melanogaster. And so we performed that same experiment, expressing a flag-tagged mosquito MT2, and then pulling it down, enriching for the viral genome for Simbus. And again, we saw more Simbus genome in the fraction that pulled down with flag-tagged mosquito MT2, suggesting that in mosquitoes, we also see a direct interaction with MT2 and the virus genome. All right, but here's where it gets different, right? And different in an interesting way. Unlike what we had observed in the fruit fly, Wolbachia actually decreases MT2 expression in the mosquito. What you can see here is relative MT2 expression in the salivary gland tissues, and I'll say it's also the same in the whole organism. For Wolbachia infected and uninfected mosquitoes in the presence or absence of virus. And the things to compare are these 2 panels. So now what we see is virus upregulates MT2 expression, and Wolbachia brings it back down. So just as we had observed in the fruit fly, virus and Wolbachia are fighting over this transcription of this one gene. It's also true in mosquitoes, but in the opposite direction. And just so I confirm, you guys, it's the Wolbachia does block Simbus virus in mosquitoes, and that's these data here. The other major difference is that mosquito MT2 is proviral. So virus increases its expression. And if we express MT2 without Wolbachia around in mosquitoes, we see infectious virus titer go up and specific infectivity also go up. So it seems like the activity of MT2 in mosquito is actually promoting virus replication and increasing its infectious virus titer. So in conclusion, mosquitoes and flies are not the same in both of these systems, MT2 is important for virus replication. But in mosquitoes, it acts in a pro-viral manner. In both of the systems, Wolbachia and virus seem to be altering the expression of this important gene, but in different ways. So we say mosquitoes and flies. Mosquitoes can fly, flies can't mosquito, but they can both Wolbachia. So all of these data lead us to ask the following two questions. So in both of these dipterans, is the virus genome modified by Wolbachia? Because that would be the natural conclusion, right? I've shown you that MT2 binds the virus genome. I've shown you that the genomes or the viruses that are coming out of Wolbachia infected cells are unable to form another infection. Maybe that's because the genome itself is modified. Could we then detect changes in M5C signatures in the virus genome? So the first experiment I'll show you, it really proved that RNA is the target of Wolbachia-mediated inhibition. So we started with mosquito cells with and without Wolbachia and produced progeny virions from them and used those to infect naive vertebrate cells. And these are tagged with this nanoluciferase reporter construct, so we can use a luciferase assay to track infection over time. And so those are these data here. So you can see across the time course of infection, when Wolbachia is present in the progeny, from the progeny virions, or they were derived from Wolbachia infected cells, we see that infection rates, or the rate at which this virus produces protein, is much lower than when they're derived from Wolbachia free cells. And you can say like, oh, well, that's just pathogen blocking, but these are naive vertebrate cells. There's no Wolbachia here anymore, right? So even if we control for infectious progeny viruses coming out of here, you see a defect in their ability to replicate in vertebrate cells, suggesting that there's something wrong with these viruses. And then you might say, well, maybe it's entry. Maybe there's something wrong. They can't get in to the vertebrate cells. Okay, so we can fix that problem by extracting total virion RNA and transfecting it into these vertebrate cells, bypassing entry. So if the defect was entry, now we should see growth the same for both the blue and the red lines. And instead, we see an even more pronounced defect for the viral genomes that are coming from Wolbachia-infected virus cells. So they carry this signature of the environment in which they were reared. It's almost like an epigenetic effect. They remember that they came from Wolbachia-infected context, and now they don't replicate as well in vertebrate cells. So that led us to ask whether we could detect M5C modifications in the genome. So we have mosquito cells with and without Wolbachia, and we took progeny viruses from both of these, extracted virion RNA, and then used LC-MS to detect nucleotide modifications with standards. And this was done in collaboration with the Zahar lab at Washington University in St. Louis. This is all done in mosquito cells. So remember that in the mosquito context, it actually suppresses the MT2 expression levels, whereas an MT2 is pro-viral. So our expectation is that in presence of Wolbachia, we would see fewer methyl marks. And that's indeed what we observed. So there's a significant effect of Wolbachia on detection of M5C in these viral genomes. And as a control, I'm just showing you M6A. There were a couple of other controls we threw in there as well, and we did not see a significant difference in the presence of Wolbachia. And this is really exciting because it suggests that there really is some change in the epitranscriptome of the viral genome that is induced by the presence of Wolbachia. So in conclusion, I've showed you that Wolbachia infection dramatically changes the host cellular environment, including MT2 expression. The MT2 is antiviral in Drosophila, but it's proviral in the mosquito, and that Wolbachia's alteration of MT2 expression affects the RNA virus epigenome. So what's next for this project? We have been lucky enough to get a Rules of Life Microbiome Theory Mechanism Award. And our collaborative group here includes Dr. Julie Dunning-Hottop of Maryland, of course, my longtime collaborator, Rich Hardy, and introducing Doug Rush of the CGB. We're going to use nanopore sequencing to directly sequence the RNA coming from these infections and really identify what the landscape of viral modifications is. you might imagine, for example, that it's kind of arbitrary, that you just see like across the board an increase or a decrease in these methyl marks. It's not really specific. Or you might imagine that they are really constrained to a particular part of the viral structure, that maybe these marks are detected by some reader that has something to do in the cell with the particular structure of the virus and how it interacts with that reader. So I'd like to thank the members of my lab that contributed to this work. I've highlighted in red our Wolbachia team. The people the work I mostly presented here was from Tamanash Bhattacharya and Amelia Lindsay, the people I highlighted earlier. Kathy Sheehan, Marion Martin, Danny Rice, and John Massey are our Wolbachia team. I thank our collaborators and then our sources of funding. And with that, I could take your questions.
Awesome. Thank you, Irene. Yeah, so we can go ahead and open it up to questions. And if we can start, if there's a brave student or postdoc who wants to start us off, we can go there. It looks like Monica has a question in the chat, which I'll go ahead and read out. Thank you for the amazing talk. Love choose your own adventure concept. Is Wolbachia does Wolbachia infect other invertebrate taxa besides insects? And is there any analog of Wolbachia widespread infection in other groups of animals? For example, other microbes that widely infect mammals or amphibians, etc.
Yeah, so I guess Wolbachia does infect other arthropods. So it infects spiders, it infects isopods, it infects filarial nematodes too. So there's a broad group of invertebrate taxa that Wolbachia infect. And a real interesting question is to think about why Wolbachia is limited to certain groups and what particular host contexts are required for infection. Is there an analog of Wolbachia? So Wolbachia is I used to start off talks by saying like Wolbachia is the great pandemic, but it seems like too soon, I shouldn't call it. Nobody likes that word. And it is, Ed Yong's favorite pandemic microbe, but there is a recent discovery by Rebecca Biga-Thurber of this rickettsia-like symbiont in marine animals. And that's fascinating to me for two reasons. One, rickettsia are related to Wolbachia. So we might have like a Wolbachia of the sea pandemic also, which would be interesting to explore further. And that's the only one I know of that might be as widely prevalent in an environment compared to Wolbachia.
Awesome.
I hope that answered your question, Monica.
I was going to say, if there are other questions, you can unmute yourself or use the chat window. Anything's fine. There's one student question, so I'll ask the question I was wondering about. So one was, I guess it's a straightforward one, and maybe I missed it. Have you looked at any like host RNAs, whether they're also being modified?
Oh yeah, that's a great question.
Whether that's different in infected versus uninfected.
Yeah. So part of this, I did show you that Wolbachia infection and Symbis alter splicing, right? So we already know that like the isoforms that are used are changed in the context of these infections, but whether they're also like if the epitranscriptome of the host is modified in a particular way, and if that changes function, that's fascinating to look at too. And that's definitely one of the goals of the grant, is to see like broadly, do we see big changes in the methylation profiles of host RNAs, because really nobody's looked, and it's always been assumed in dipterans that it's tRNA as the target because that's the only experiment that's been done. So methylation of tRNAs alters their stability.
Okay. Yeah, I guess that gets to something else I was thinking about, which is people think about all the effects that different Wolbachia can have on, and it depends a lot on Wolbachia strain and host genotype. But when you start thinking about something broadly like RNA modification, I mean, I wonder if that might help explain some of the like host genotype dependent effects that you see, right? If there are different targets, if there's effects on transcriptome in the host, and that depends on host genotype.
Absolutely, yeah. And understanding why it is that in these two insects there's such a very different response. I mean, it is both through MT2, so that's interesting, but the fact that Wolbachia puts it one way in one host and one way in another one is fascinating too. We don't know how we get to the transcriptional level, like what is the mechanism by which Wolbachia infection gets to changing MT2 expression. So that's something we're actively trying to figure out now, but yeah.
other questions?
It was just so clear.
All right. Well, if there isn't anything else, I think let's thank Irene one more time, or virtual clap, or real clap. And I believe according to the schedule, we'll take a few minutes break, and then people can feel free to stick around. We have our like virtual tea, coffee, Q&A session starting about 1:15 usually is when we, is that right, Peter, when we start? Okay, cool.
All right.
All right.
Is it the same room or no?
We're just gonna stay, yeah.
Okay.
So you can
Cool.
like